Vision and you, how does it work? Today you'll learn about vision by Lindsey and Stile Wreggelsworth. How do we actually see with our eyes? When we look at an object, light bounces off and reflects off the object to travel into our cornea and lens. The cornea and lens are features of the eye that allow us to focus the light and make objects look clear. Once light travels through the cornea and lens, it will hit the retina, which is located in the back of our eye. The cells within the retina is the start of what processes the light into a neural signal that our brain can perceive as an image. The cells within the retina are known as light detecting cells. The two types of light detecting cells we have are rods and cones. If you are in a dark room, rods will be turned on. But if you are in a room with the lights on, cones will be turned on. Since objects come in different colors, certain cones are turned on and activate their photopigment cells to perceive that color. There are only three types of cones. One for when red, green, or blue light hits our eyes. If we only have three cones to see only three colors, how are we able to see colors like yellow? If we want to see yellow, you will notice that yellow light will reflect off the banana into our eyes and it will turn on red and green photopigment within the cones. The red and green photopigment within the cones then mixes their signal down the optic nerve and signals to the brain for the eyes to see yellow. This can be compared to mixing colors with paint. If you only have red, green, and blue paint and want to get yellow, you must mix a certain amount of red and green to make yellow. Do our eyes ever get tired of seeing in color? Of course they do. Let's find out how this happens. Stare at the white spot in the middle of the star for 6 seconds. Then stare at the white page next to the star. You should see that the purple on the outer ring of the star is now where the blue is, and the blue is now where the purple is. This is the concept of after images. This occurs when the cones within our eyes become fatigued and produce an inverted set of colors in an image that we are staring at. An example of this is shown on the right. So what's it like to really be colorblind? When someone says they're colorblind, this does not mean they cannot see colors. What it actually means is that people who are colorblind can only see certain shades of colors. There are multiple forms of color blindness. The type we'll be talking about is protanopia. Thinking back to a few slides ago, we talked about how red cones contain red photopigment. But if you are born with protanopia, then the red cones in your eyes are filled with green photopigment. This causes a person to not be able to tell the difference from red and green. You can see what an individual with protanopia sees by looking at the pizza and Pepsi logo. Any sauce on the pizza appears green to them, and the red on the Pepsi logo appears green as well.
So, how do signals enter the brain? Well, once the rods and cones have been activated, they then leave the eye through the optic nerve. The optic nerve then splits its signals to different sides of the brain. Which path the signal takes depends on where on the retina the light hits. The light that hits on the inside of your eye, near your nose, crosses over to the opposite side of the brain. The light that hits on the outside portion of your eye, towards your ear, proceeds on its path on the same side of the brain. Regardless of which side the signal goes, it always passes through the lateral geniculate nucleus and then on to the primary visual cortex. The point where the signals cross over is called the optic chiasm. Now let's do something fun. In the previous slides, we mentioned the optic nerve. Well, the point at which the optic nerve begins is the point where your eye has no rods or cones. Therefore, each eye has a blind spot. You can see your blind spot by looking straight at the image with your nose positioned somewhere between the plus and the circle. Now close your left eye and focus on the plus sign with your right eye. Do not look deliberately at the circle. Now move closer to the image, slowly. Don't take your focus off the plus sign while you are doing this. At some point between 10 and 14 inches, the circle will disappear from your peripheral vision, and the brain will read the surrounding white color to fill up the empty space. This is your blind spot. Cool, right? But does everything have a blind spot? No, they don't. While this is true for most animals, cephalopods, like the squid and octopus, have no blind spot, as they have their photoreceptors in front of their supporting cells, which is the opposite of the way that humans work. Thank you for listening. Anybody have any questions?